Yes, so today I'm going to talk about clonal repertoire analysis of B cells and T cells. So because this topic is very much within immunology, and because I'm quite certain not everyone has a background in immunology, I'm going to spend most of the time talking about the biology of B cells and T cells and their receptor repertoires, and then go into how exactly we go about processing BCR and TCR repertoire data. So, the types of cells that we're going to spend this lecture talking about are B cells and T cells. So B cells are the first thing that we're going to talk about, and these are critical workhorses of the adaptive immune system. And that is because they are the cells that produce antibodies. Now, antibodies are proteins that are released into the blood and mucosa, and they provide protective immunity by binding to structures on the surfaces of pathogens called antigens. So they have a protein structure that happens to match structures on the surface of viruses or bacteria, and they fit together like a lock and a key. And they can neutralize pathogens by just coating their surfaces and preventing them from interacting with cell receptors, or by activating other parts of the immune system to destroy them. So that's how antibodies should work. However, it's also possible that antibodies can bind to self-antigens, and this can cause autoimmune diseases. Certain types of antibodies can bind to otherwise harmless food antigens, and this can cause food allergies. And B cells seem to have a dual role in cancer response. So some B cells can bind to tumor antigens and provide anti-tumor immunity. However, there are some studies that suggest that B cells can sometimes play an almost pro-tumor role in cancer responses. And B cells themselves can become cancerous by turning into B cell lymphomas. So B cells are a source of great power within the immune system, both for good and for bad. We're also going to talk about T cells. So T cells come in two flavors. The first is CD4 T cells. And T cells help to coordinate immune responses and to kill infected cells. So one of the main roles of CD4 T cells is to activate B cells and coordinate the adaptive immune response so that you get effective antibodies produced against viruses. They can also release cytokines that activate parts of the innate immune system that can directly consume pathogens. And they can help to activate CD8 T cells, which are the other main type of T cell called cytotoxic T cells. And they can destroy virus-infected cells and tumors. So T cells play a diverse set of roles within the adaptive immune system in conjunction with B cells. So just to start at a very broad view of the immune system, there are two main wings of the immune system. We're going to spend most of the day talking about the adaptive immune system, but it's important to understand the innate immune system as well. So the innate immune system is the body's first line of defense once the physical barriers like the skin or the mucosal membranes have been breached. It's rapid, non-specific, and typically triggers inflammation. So this is composed of cells that are just in the site of infection, usually there at the time of infection or recruited soon after. It's composed mostly of macrophages, which are large cells that endocytose bacteria and viruses, or neutrophils, which can also endocytose invading pathogens. They can also undergo this really interesting process called netosis, where they basically explode their DNA onto bacteria and kind of net them up. That was a recently discovered thing. And also natural killer cells, which can make infected cells undergo apoptosis. So this is the innate immune system, and it's all geared towards rapidly responding to infecting bacteria and viruses. And then there's the adaptive immune system. So this activates if the innate immune system can't handle an invading bacteria or virus. It's slower to activate, but it is specifically targeted to the threat that is detected. It's composed, as I said before, of B cells, which make and release antibodies, CD4 T cells, which coordinate cellular responses, and CD8 T cells, which destroy infected cells. While most of immune repertoire analysis focuses on adaptive immune cells, it's worth looking at how innate immune cells work. So these recognize conserved molecular patterns of infection, like bacterial cell walls or cytoplasmic DNA or free-floating RNA. This works by having receptors that are encoded within the genome and specifically recognize these patterns. So we have genes within our genome that encode receptors that recognize highly conserved molecular patterns. Classic examples are the bacterial cell wall that you find in gram-positive bacteria. That activates what's called the toll-like receptor 2, and that initiates a set of pathways that activate inflammation. There's also lipopolysaccharide, which is a very conserved molecule on the surface of gram-negative bacteria, and that activates the toll-like receptor 4 pathway, which is found on a lot of innate immune cells, and that activates cytokines and type 1 interferons. Basically, these allow the immune system to recognize when a gram-positive or gram-negative bacteria is present and then activate the immune system. But importantly, it works by focusing on these highly conserved patterns that are very classically associated with bacteria and not human cells. And it's pathogens that we have evolved with for a very long time. One issue with pathogens, though, is that pathogens evolve much more quickly than we do. So a classic example is SARS coronavirus 2, which is the virus that causes COVID-19. That emerged in 2019. So we don't have inbuilt receptors that recognize patterns on SARS coronavirus 2 because it emerged only very recently. And in the last 5 years, it's created this huge, diverse family tree of variants that are able to be more infectious or evade our immune system, so like Delta, Omicron. So this is kind of a conundrum and really a vexing problem in immunity, which is we as big multicellular organisms evolve much more slowly than the viruses and bacteria that infect us. So how exactly is it that we are able to create an immune response against something that evolutionarily we just haven't seen before? Any guesses? What's that?
Rearrangement of the genetic rearrangement, basically we create more random subsequences to
Right. Yep. So there's a common process of the adaptive immune system, and that is starting with progenitor cells, where you have random genetic rearrangements of receptor genes. This creates a large pool of naive cells with unique receptors. And then once we have this large pool of naive cells, we then select those cells for expansion and production of memory cells based on the pathogens we're actually exposed to. So it's this process of first generating a large, diverse pool of random receptors and then selecting the ones that actually bind to viruses. And this allows us to anticipate and learn patterns of the pathogens that we're actually infected with. So I'm going to first talk about B cell receptors and then move on to T cell receptors. So B cell receptors are essentially membrane-bound structures of antibodies, and they're composed of a heavy chain and a light chain. As you might expect, the heavy chain is larger than the light chain, and the antigen binding region is usually depicted here, and is composed of the complementarity determining regions of both the heavy chain and the light chain. Each B-cell receptor has two antigen binding regions, though antibodies, when they're released, can have more than that. And this is the usual structure of secreted antibodies. Similar to the BCR, there is the variable region, and that determines the binding target. And there is the constant region, which, as the name suggests, also varies, but not as much. And that determines what the function of the antibody is when it is bound. Okay. So each B cell, or each naive B cell that is produced, produces a single type of a randomly produced receptor. So the way that B-cell receptor repertoire diversity works is that gene segments are randomly rearranged within new naive B cells. And this produces a large diversity of naive B cells, each expressing a more or less unique receptor structure. So we have B cell 1, and it has these different gene segments for its receptor, B cell 2 and B cell 3, each with different gene receptors. And of the about 10 to the 11 B cells in your average human adult, there's more than 10 to the 14 possible receptor combinations. So this is a really huge diversity of potential receptors, and in fact, larger than the body actually has to produce. And this large, diverse repertoire can recognize a vast array of proteins. More specifically, so we're going to focus on the heavy chain first because it's the most diverse and generally the more important one for binding. So the heavy chain locus in humans is composed of multiple variable diversity and joining segments, which are called V, D, and J segments. It's located on chromosome 14, and each time a new naive B cell is produced, 1D and 1J are spliced together, and then this complex is spliced to a V gene. And this creates a permanently rearranged genomic DNA within the B cell. So the genome of this cell actually changes, and these intermediate D genes and J genes are cut out here, and these intermediate V genes are cut out here. So if you were to sequence the DNA of this cell, it would be different from most other B cells. This can then be expressed and processed to give us the processed BCR mRNA within that cell. And just a bit about the diversity of these gene segments. So most of the nucleotides are found in the V gene. They range between 165 to 300 nucleotides, although most of them are more than 200 nucleotides. So within a BCR, most of the sequence nucleotides are actually in the V gene. D genes are a lot shorter, so short that it's actually kind of hard to tell what is a D and what is just other parts of the sequence. We'll get to that a little later. But it's really important for diversity of antigen binding regions. And then there is the J gene, which is the least diverse of all of the gene segments and is mostly there for structural integrity of the molecule. So in addition to the combinatorics of finding different V genes, D genes, and J genes, there is also what's called junctional diversity, which is created by the process of stitching these different gene segments together. So the way these are joined together is through what are called N and P additions. So it starts off with what are called P or palindromic nucleotides. This is because they are the same going forwards as backwards. So they're caused by some nucleotides occurring and then attached to their reverse complement. This is because they're initially formed by hairpin loops that open up and then have these ragged edges. So starting with these palindromic nucleotides, there are enzymes that add what are called non-templated nucleotides, so just random additions of nucleotides. And that occurs until there is a sufficient match between these two segments. Once that happens, the two DNA strands join together, and then the rest of the junction is filled in by reverse complement. So we start off with these palindromic nucleotides, and then we have random additions until there is overlap, and then we can fill in the rest by reverse complement. So a few things about this process. One, it's highly random. So these are nucleotides that are just kind of added in between V and D segments and also between D and J segments. And it also allows for length diversity of gene segments. So this doesn't necessarily occur over a specific sequence length. So these junctions can range in size. The largest we see, I don't know, maybe 30 amino acids, so maybe even larger. So this vastly increases the kind of diversity that BCRs can be produced. And importantly, this junction region is the most important antigen binding region of this DNA. Okay. So in addition to the junction region, there is also the light chain, which, so to form a productive BCR, we need a heavy chain and a light chain. And the light chain undergoes a similar process of somatic recombination. Except for the light chain, there is generally less diversity, so fewer gene segments, and also there's no D gene. So for the light chain, you just have V genes, and they splice to a J gene. And because of this, there is a lot less diversity in the light chains that are produced. However, the light chain is required for the BCR and antibody to function, and not all heavy chains can pair with all light chains. Importantly as well for humans, there are two light chain loci, Ig kappa and Ig lambda. I don't know why the heavy chains are in English and the light chains are in Greek, but immunology is weird like that. Okay. So fitting this all together, this diversity of BCR sequences generates a large diversity of protein structures. So both chains contain what are called framework and complementarity determining regions, and these regions are non-consecutive. So if we look at our V, D, and J regions, the first framework region occurs at the beginning of the V gene, then the first CDR, then the next framework, and so on, until we get to the CDR3, which is composed of the NNP additions in the junction, as well as the D gene, and then the remaining NNP additions. And then finally, we have framework 4 in the J gene. The reason these are non-consecutive is that when the protein forms, it folds over so that the framework regions connect to each other, and that gives the molecule stability, and the complementarity determining regions stick out. So after this peptide folds, so this is the heavy chain, it pairs to a light chain, and then you have two different subunits pairing together. And this gives you the complete expressed BCR on the cell membrane. And this part of the receptor is where antigens bind. So this is how we go from DNA rearrangements to protein folding to having a complete BCR on the cell surface. And this whole process repeats each time a new B cell is produced. And this is how B cells are so diverse. But wait, there's more. So after B cells are activated by binding to some kind of antigen, they undergo an additional process called somatic hypermutation and affinity maturation. Within this, so when B cells are activated, they enter into what are called germinal center reactions, where they actually upregulate enzymes that damage their DNA. And this DNA damage produces mutations, some of which improve the binding affinity of their BCRs and some of which decrease the binding affinity of their BCRs. The cells with mutations that improve their binding affinity survive and reproduce more than the cells with lower binding affinity. So this is an evolutionary process of mutation and Darwinian selection, which leads to greater diversity of BCRs and really fine-tuning of the B cell receptor to binding to whatever antigen is present. And this leads to a much higher affinity of the antibodies that are released. So we can think of VDJ recombination as these random guesses in antibody space. And those random guesses allow the immune system to recognize essentially any virus or really anything out there. However, when we have an immune response, we don't want random antibodies. We want antibodies that are very fine-tuned and targeted towards the viruses that we're actually exposed to. And this process of affinity maturation is that fine-tuning. It really allows the B cells to find mutational variants and sequences that neutralize viruses. So all the variation I've talked about so far has been in the variable region, which controls what an antibody binds to, but there's also variation in the constant region. And this encodes what, or it affects what effect the antibody has once it is bound. So the constant region gene encodes what are called different isotypes, which control antibody effector functions. The first isotype is IgM. So all B cells, when they're first produced, have IgM BCRs, and if they make antibodies, this is the type of antibody they produce. It's this kind of cool ninja star looking antibody, and importantly, it has 10 binding regions. So one of these antibodies can bind to 10 different antigens, and 10 of the same antigens, but different molecules. So this would have what's called high avidity. So a single antibody can mark up to 10 different bacteria or viruses. The next antibody class is IgG, and this is the most common antibody class in the blood and the serum, and it has 4 different functional subclasses. G1 is the most common, and G4 is the least common. These are generally when people talk about antibodies, this is the kind of antibody that they're talking about. So they're really good at binding to viruses and initiating immune responses to viruses and things in the blood and the serum, except for G4, which seems to have almost tolerogenic consequences. It sort of competitively binds against other antibody classes. It's weird. Then there is IgA, which has 2 different subclasses, and IgA is the most common antibody found in the mucosa. So in the gut or in the linings of the lungs or any of the mucosal membranes, the predominant antibody is IgA. And this dimeric structure allows it to survive highly acidic environments. And it's very good at clumping together bacteria. Then there's IgE, which is mostly involved in response to large multicellular parasites. And annoyingly for people in the modern world, is also the antibody that causes allergies. And then IgD, which is weird. Kind kind of a mysterious antibody. So naive B cells express both IgM and IgD. It's not always clear what the IgD is doing. I'm sure there's something, though. Importantly, because isotypes are encoded by the constant region genes, B cells can switch between different isotypes. And this allows them to target their antibody response to different parts of the body or to different types of pathogens. But because isotype switching occurs by removing gene segments, it can only occur in a particular direction. So once you've started at IgM and switched to IgA1, you can't go back to G3 or G1 because those segments were cut out in the splicing process. Okay. So, just to summarize what we've talked about, so adaptive immune receptors are shaped by diversity and selection. BCR diversity comes from VDJ recombination. If you're wondering, I only learned this recently, the D is usually in parentheses because there isn't always D recombination. So this refers to both VDJ on the heavy chain and VJ recombination on the light chain. I feel like I should have learned that earlier. NNP additions as well as heavy and light chain pairing. After activation, B cells proliferate to form germinal centers and groups of related cells called clones. And antibody binding and function is improved by hypermutation, selection, and isotype switching. Questions about any of this so far? Yeah.
It's not really directly related, but the antibodies, they're also antigens, right? So these are proteins that the immune system has never seen. So are there antibodies against variable regions of antibodies? So it never happens.
You would think that would happen, but I think generally no. You can get immune complexes forming. Generally, I don't, I think the reason we don't get immune responses against antibodies themselves is in order to initiate these kinds of activations, you need some kind of danger signal. So basically, you need the innate immune system to activate the adaptive immune system and target it towards a particular antigen. And it doesn't seem to do that against antibody variable regions. So it is a good point that you're creating these proteins that the immune system or the body hasn't seen before. But just having a protein that your immune system hasn't seen before isn't enough to mount an adaptive immune response. That's a good point. Yeah. Okay. Yeah.
Sorry, I just wanted to add that have like a much shorter structure. I just wanted to add that does that like goals like less possible varieties.
I'm sorry, I didn't.
I said canalids have like much more complex structure.
Right.
And which are used for like canalids. And I just wanted to add that does this structure like configuration goes like less possible like the complementary structures.
So do camelids have less complementary? Right. So yeah, camelids, they can make antibodies that don't have light chains. Those, my understanding is that those have less BCR diversity as a result, but interestingly, lacking the light chain, they're able to penetrate like viral glycoproteins really well. So camelids are really good at making like anti-HIV antibodies, which is quite cool. So I know camelids can also make antibodies that do have light chains. So in making antibodies that don't have light chains, it's oddly a different kind of diversity for them. So, yeah. Okay. Cool. So, on to T cells. So, as I said before, T cells come in two main varieties and a third. We'll get to that. So, the the main T cell receptor loci are the alpha and beta loci, which form what we would call alpha beta T cells. And these are somewhat analogous to BCR heavy and light chains. So the beta chain is quite similar to the B cell heavy chain. The alpha chain is similar to the B cell light chain. The main difference between CD4 and CD8 cells with regards to their receptors is that CD4 T cells bind to antigen that is presented on MHC class II, which is expressed by special immune cells, and it presents longer and complex peptides. Whereas CD8 T cells bind to antigen that is presented on MHC class I. And that is expressed by nearly all cells, and it presents shorter 8 to 11 amino acid linear peptides. So we can think of MHC class I as just something that's on all cells to express peptides that are currently being produced within them. So if cells are producing a virus unknowingly, they will chop up proteins from that virus and express it on MHC class I. And if there are CD8 T cells that recognize that antigen, they kill that cell. Whereas MHC class II is a special antigen presentation molecule on the adaptive immune system. And that's only presented by special cells that focus on activating the immune response. And then there are the gamma delta T cells. So gamma delta T cells are produced by a different locus and different rearrangements. They're sometimes called innate-like T cells because they behave more similarly to innate immune cells. They're within the tissues, and it's not really clear what triggers their activation. It seems like they don't actually need antigen to be presented on MHC. Generally, their function and role within the immune system is a lot less well known. And like most lectures on this topic, I will mention their existence and not really talk about them again. So all of what I'm going to talk about after this is just alpha beta T cells. So as I said, the beta chain for alpha beta T cells is quite similar to the BCR heavy chain. So it's composed of multiple VD and J segments. When a new T cell is produced, one of each is spliced together, and this forms a permanently rearranged genomic DNA within the T cell, and that gives you a new processed alpha chain mRNA. Similarly to the light chain, we also have the beta chain, and this has V segments and J segments, and those are spliced together to again give a permanently rearranged alpha locus within a T cell. Similarly, again, to BCRs, the TCR molecules are composed of non-consecutive framework complementarity determining regions. And the frameworks are spaced out similarly, and the CDR3 is composed of this junctional NNP additions, the D gene, and the NNP additions between the D gene and the J gene. And when we have molecules forming, the frameworks associate with each other so that the CDRs stick out, and you have this CDR3, which is especially important. And both the alpha chain and beta chain are expressed on the surface of the T cell. If it's a CD4 T cell, this would bind to MHC class II plus presented antigen. And if it's a CD8 T cell, it would bind to MHC class I. So MHC class II and class I are often described as being like hot dog buns, where the MHC class II, the hot dog sticks out beyond the bounds of the bun. And in MHC class I, the hot dog is cut so that it doesn't go beyond the bounds of the bun. I don't know if that analogy really lands outside the US, but This is how I always think about it. And some of the immunology textbooks will literally have pictures of hot dogs and buns to describe this. Okay, so again, similarly to B cells, T cells clonally expand. So there is this huge naive diversity of T cells. But when they are activated by binding to their cognate antigen and receive additional signals from other immune cells, this causes them to activate and proliferate. And this creates much more copies of the TCR that is relevant to the virus that's actually infecting us. It also creates T cell memory. Now, there are a lot of ways that T cells and B cells work together to create an immune response. But I think one of the most illustrative of how the adaptive immune response requires both TCRs and BCRs to work together to mount an effective response is in what are called germinal center reactions. So these are reactions that occur between B cells and T cells and dendritic cells. Typically, they happen within lymph nodes, and they have the goal of creating neutralizing antibodies against a particular antigen. So let's say you have a virus that is infecting you, and it is coded in this yellow square antigen or yellow diamond antigen. So if we have a B cell that binds to that antigen, say this is a naive B cell, it doesn't bind very well, but enough to capture it, it will digest this antigen and present it via MHC2 to a CD4 T cell. And if that T cell is activated, it will bind to a molecule on the B cell called CD40, and this will cause the B cell to activate. So T cells can go off and expand and form clonally expanded T cells as well as memory B cells, and the B cells enter into germinal center reactions within lymph nodes. And germinal center reactions are composed of a dark zone where you have somatic hypermutation, so this process of rapid mutation and proliferation, and the light zone where you have selection for antigen binding. The way this works is you have dendritic cells, which are these cells that have long arms that are coated with antigen. And when B cells emerge from the dark zone, where they've mutated and they have these slightly varying B cell receptor structures and affinities, they will compete against each other to try to pluck as many antigens as possible off of the surfaces of dendritic cells. They then present those antigens to T cells within the follicle, and they receive survival signals proportional to how many antigens they present. This means that the B cells that are able to bind to more antigen get more survival signals. So they enter back into the dark zone where they proliferate and mutate more. So this is the mechanism of how we get clonal expansion and mutation and increasing affinity of B cells. And importantly, the cells that bind less well die. So the default state of B cells in this process is apoptosis. After some time, this produces mutated memory B cells. So these B cells are quiescent and they can hang around in the lymph nodes, but also in the blood. And if you're exposed to this same pathogen again, they will spring back into action. It also produces what are called plasma cells. Some of these cells are short-lived and they just produce lots of antibodies in a short period of time. And some of them are long-lived plasma cells, and they migrate to the bone marrow where they release lots of antibodies for decades. And these antibodies, because they've undergone this germinal center reaction, are neutralizing and class switched, and they go out into the blood and mucosa and do violence against whatever has that antigen. Okay. So to summarize part 2 of this, TCR diversity forms from similar VDJ recombination as in B cells, and T and B cells work together to orchestrate germinal center reactions. So the next section is focused on the question of how exactly do we make sense of all of this diversity of TCRs and BCRs and use that to understand how the immune system is changing in people or in model organisms. So the kind of data that we use to study immune receptors is called adaptive immune receptor repertoire sequencing, or AIR sequencing. In this, we sequence the BCR and TCR genes. This can give us millions of sequencing reads for bulk BCR approaches or tens of thousands of single cells for single cell sequencing. At least that's the current technology. It wouldn't surprise me if in the future we're able to go an order of magnitude higher. Importantly, for single cell sequencing technologies like 10x 5' VDJ data, we can pair BCR sequences with RNA-seq, and this can tell us about gene expression information for different B cell receptors. So we can associate individual BCRs with different B cell and T cell subclasses. Currently, single cell sequencing technology is changing rapidly and giving us ever more ability to associate biological information with BCR and TCR sequences. So chief among these is single-cell RNA-seq, but there's also site-seq, which can tell us about cell surface markers, and of course, spatial RNA-seq, which allows us to pair spatial coordinates with RNA sequencing data. So these new technologies can give us unprecedented resolution of individual cells, and also the BCRs and TCRs that are expressed by those cells. So there are multiple steps for adaptive immune receptor repertoire analysis. The first is sequence quality control and preprocessing. So this is underappreciated but vital. I would say that good sequence preprocessing is the difference between looking at biological signal and biological noise or technical noise. So AIR sequencing can have a very high error rate, and there are multiple approaches to correct this error. That tends to come from either PCR amplification or sequencing error. One of the most important is using what are called unique molecular identifiers, or UMIs, and this allows us to map different sequence reads to the same original molecule. So we can create what are called consensus reads, and this allows us to correct sequencing errors or PCR errors. The way this is done in practice is that many sequencing technologies like 10x have their own proprietary preprocessing software, and it's generally recommended to use those because they're specifically designed for the technology that they're used for. For everything else, like bulk BCR sequencing, we tend to use Presto, which is a toolkit developed by the Kleinstein Lab. And it's a very flexible way of assembling consensus reads and doing primer alignments, basically doing everything that you would need to do in sequence preprocessing and quality control for BCRs and TCRs. The next step is identifying the V genes, J genes, and constant region gene segments in the BCR. The way this is done is by aligning BCR or TCR sequences to a reference database of germline alleles. The most common database is called the IMGT database, or the Immunogenetics database. There's also the Open Germline Receptor Database, or OGRDB, that's growing and becoming more popular. And basically, you combine these germline gene segments with programs that align your sequence to those sequences. And you can then use that information to tell which V gene, which J gene, and which constant region your sequence uses. Generally, the junction region is hard to figure out by this approach. So it is sometimes possible to identify the D gene based on alignment to a reference germline database. However, because D genes are very short and diverse, those alignments are often unreliable, and we generally ignore any assignment of D genes by one of these alignments.
Can I ask a question?
Sure.
So the VDJ construct, like the resultant gene is quite long, right? Do you always use long read sequencing, or you have to sequence through the whole, you have to get this sequence of the whole thing, right?
Right. So you could use a couple of approaches. You don't need to use long read sequencing. So the full length of the heavy chain is usually like 400 base pairs. So it's possible to use short read sequencing with paired ends to get both sides of the sequence and then assemble them based on that.
The overlap.
Yes. Yep. So most Illumina sequencing can give you 250 base pairs for each read. So there's usually enough overlap to assemble the molecule. Older approaches, you didn't always get the overlap, and you had to just sort of have a gap in between. Not ideal. Of course, the full sequence is a lot more important for B cells, because with B cells, we often are interested in the somatic hypermutations. Because T cells don't hypermutate, oftentimes you really just need enough of the enough of the V gene to assign the V gene sequence, and then the junction sequence and the J gene. But yeah, you can use short read sequencing, and that works fine. Long read BCR is mostly used if you want to get the full constant region as well.
The other question is, so when you talk about clones, do you conceptualize clones as a unique sequence of VDJ, or you can have the exact same VDJ arrangement, but then you see difference in the junctional region, and this makes the difference?
Yeah, so there is differing ways of assigning terms to biological entities with this. Generally, this probably comes from working primarily with B cells. I would say I would consider a clone to be a set of cells that descend from a common VDJ arrangement. So in B cells, that would be BCRs that have hypermutations that potentially differ from each other, but ultimately descend from a common rearranged naive B cell. And I would say that also extends to T cells. For T cells, because they don't undergo somatic hypermutation, in practice they end up being the same BCR. But yeah, I'll get to how we do clonal clustering in a bit. But okay, so this is the junction. The N and P nucleotides, of course, because they're random additions, cannot be identified by alignment to a germline database. We can identify where they are, more or less. We can, however, reliably identify the junction sequence because it occurs at very conserved regions in the V gene and the J gene. And generally, when we do the alignments, the notation that's used is standardized so that it shows the locus. So here IGH means heavy chain. The family, so the family here would be V1. The gene would be V1-18. And the allele, so this would mean a genetic variant that's segregating within the human population, would be 1. So this asterisk and then 2 letters is to identify different alleles of these different gene segments. And there's a similar notation used for the D gene and the J gene. And the constant region here is shown as IgM. Normally, we don't identify constant regions using IgBlast. We would use that in pre-processing by alignment to primers, but it can also work this way. One important concept for BCR and TCR analysis that is a little hard to get at first is what's called IMGT unique numbering. So this is a numbering and alignment system that is developed because CDRs and frameworks occur at consistent places within BCR and TCR sequences. And this numbering or alignment scheme adds dots to the alignments so that the CDRs and frameworks occur at specific locations. So within this IMGT numbering scheme, the framework 1 is always from position 1 to position 78. And CDR1 is from the next nucleotide to position 114. The CDR3 is a little bit weirder, so it goes from a conserved cysteine residue at codon, well, it ends at nucleotide 312, and then it goes to a conserved tryptophan or phenylalanine residue in the J gene. And then the last framework region goes from that residue until the end of the sequence. So this is a way of, given a particular BCR with a set of V gene and J gene genes, you can place that BCR in a common alignment system so that the regions of that BCR always occur at consistent places. So if this is our input sequence, and this is the 54th nucleotide, and this is some number of nucleotides later, if we place that sequence in IMGT gapped alignment, these dots are added within the CDR1 so that just looking at the sequence, we can say, well, we're starting at position 54, and we're going out to position 78, that's framework 1. And then from position 79 to 114, that's CDR1. And then beyond that is framework 2. So importantly, if you're looking at BCR sequences versus aligned sequences, look for these dots. And if you see these dots, that means that the CDRs and frameworks occur at consistent places. Yes?
Is it possible for CDR sequences to overflow this numbering due to junctional diversity?
I'm sorry, could you?
Is it possible for CDR sequences to overflow these coordinates due to junctional diversity?
Right. So the original way of doing IMGT numbering also added these dots and numbering schemes to the CDR3. In most modern usages, that is not done because there is no actual limit for how large the CDR3 can be. There's probably some biological limit, but if you were to propose a maximum sequence length for the BCR, I'm confident someone somewhere would find a BCR sequence that would exceed it. So generally, we only add the dots to the CDRs 1 and 2. And that's because those are conserved compared to the CDR3. But they occur within this germline database and in known positions. But yeah, that is a good point, and one reason why the IMGT numbering scheme has changed a little bit in recent years. Cool. Okay. The next step is identifying B and T cell clones. So these are cells that descend from a common VDJ arrangement. Typically, we group cells by V gene, J gene, and junction length. So these are things that are formed when a VDJ recombination event happens. T cells should have the same junction, maybe a little bit of diversity due to sequencing error. But B cells are allowed to have junctions that are different. So for that, we group B cells by junction similarity. And this, of course, creates a classic problem in bioinformatics, which is how big of a difference is enough? And there are many answers to that. The way that we tend to do it is by visualizing the distance of each B cell to its nearest non-identical neighbor. And if you do that, you often get a bimodal distribution, which is interpreted as differences within a clone and differences between clones. And we can set the threshold by finding the minimum between these two bimodal distributions. So this is important because it tells us which B and T cells arose from expansions during immune responses. We can quantify these clonal expansions using diversity statistics. And this tells us the degree to which B and T cell clones are related. So higher diversity indicates lots of small clones, low diversity indicates more larger clones. And it's important to adjust for sequences and sequencing depth among samples. I'm going to move through these a little bit quickly since I'm running out of time. Another important thing that is unique to B cells is quantifying B cell somatic hypermutation. So for this, we would just compare the sequence that we observe to the germline alignment. And because the germline alignment for the junction region is always a bit dicey, we only really look at the V gene segment. So from IMGT positions 1 to 312. And we represent the somatic hypermutation as just the normalized Hamming distance between those two sequences. We can use these mutations to build what are called B cell lineage trees. And this is one of the main focuses of my own research. So in these trees, the tips are observed BCRs and the branch lengths are somatic hypermutations, leading from each tip back to the germline. And a lot of what we currently do in my lab is using these trees to understand immunological connectivity between tissues and how B cells are moving between them, how B cells are transitioning between different cell types, and how they're evolving over time when we have longitudinal samples over time. With single cell data types, we can use cell barcodes to match a lot of rich other types of biological information to BCRs. So here we have B cells that are annotated by cell type from single cell RNA-seq. We can visualize this by building B cell lineage trees and then plotting the cell subtypes for each of the cells at the tips. So here we have a lot of tips that are germinal center B cells, and here we have some tips that are plasmablasts, shown here in red. So just to summarize this, error analysis is all about translating sequence diversity into things that we can understand. Clones, lineages, things like that. The typical workflow is pre-processing, alignment, clonal clustering, and then quantifying diversity, somatic hypermutation, and if you're studying B cells, building lineage trees. The main way we recommend approaching error sequencing analysis is through a suite of programs called Incantation, which is a start-to-finish set of BCR and TCR analysis programs. It's a multi-lab collaboration, but led by the Kleinstein lab at Yale. It's modular and been adapted towards new BCR and TCR data types for the last 10 years. And we place a really high premium on documentation, helping users, and version control. So we really try to make it as usable as possible. And the R package that my lab focuses on specifically is called Dowser, which is a general purpose BCR analysis toolkit for lineage tree analysis. Some of the things that we've focused on within Dowser have been methodological advances for building B-cell lineage trees. So those have been mostly focused on building B-cell lineage trees that account for the unique biases of somatic hypermutation. So if we think about phylogenetic methods, those have been around for 100 years or so and were designed to study things like squirrels evolving over millions of years, right? That's very different from the types of mutations that you see in cells that are enzymatically mutating over several weeks. So we developed lineage tree-based methods for that. Also, methods for understanding B-cell evolution over time using longitudinal data, understanding cell migration and differentiation if we have cells that are sampled from different biopsies or different B-cell types. And we also do a lot of collaborations with experimental groups. So we've done one study recently in tracking the Moderna response to the Moderna COVID-19 vaccine, analyzing how lymphoma driver mutations hijack B-cell evolution, and in identifying the origins and causes of antibodies that cause food allergies, specifically peanut allergies. So I'm a little over time, but thank you for your attention. As I said in my introduction slide last night, or my shot talk, my lab is located at Dartmouth here in New Hampshire, and this is a much larger photo of all of us. And of course, it includes Oreo, which is our lab mascot. So, thank you.